My name's Connor, and my proposal is for an investigation into the effects of the cyanotoxin BMAA on amino acid metabolism in vitro, supervised by Ken Rogers and co-supervised by Alan Faith. Some neurodegenerative diseases occur due to proteinopathies, or irregularities in protein form and function, such as protein aggregation. Protein aggregates can form when factors that affect protein expression cause structural changes in proteins that cause them to lose function and disrupt neuronal architecture of the nervous system, further affecting the biochemical pathways of the nervous system, such as through intercellular transport disturbances, and leads to loss of neuronal function and cell death. Motor neuron disease, or amyotrophic lateral sclerosis, ALS, is a neurodegenerative disease characterized by upper and lower motor neuron degeneration that can cause dementia, muscle weakness and spasm, tremors, impaired speech, chewing and swallowing, and slow movement. Approximately 90% of cases occur at random, and 5 to 10% of cases are familial or inherited. There are also instances of clustered cases, such as those discovered afflicting the indigenous Comoro people of the western Pacific island of Guam. The incidence rate of a unique presentation of motor neuron disease called amyotrophic lateral sclerosis, Parkinsonism dementia complex, or ALS-PDC, was discovered to be between 50 to 100 times more common than in the general population. Through investigations performed since its initial epidemiological discovery in 1945, the cause has been narrowed down from a plethora of environmental factors to a single amino acid from the cycad species of a plant called BMAA. This amino acid was found to be in its highest levels within the plant in a protein-associated form in symbiotic cyanobacteria of the colloid root of cycads. BMAA, or beta-methylaminoalanine, is a non-protein amino acid. That is to say, it isn't one of the 20 canonical amino acids used by the body to synthesize proteins. It's a derivative of the canonical amino acid alanine with a methyl amino group on the side chain. BMAA exhibits an acute excitotoxic effect by stimulating metabotropic glutamate, NMDA, AMPA, and kainate receptors, as well as causing increased oxidative stress. The property of BMAA of most interest to this project is that it can form reservoirs of toxin in organisms by bioaccumulating in the food chain. Early research excluded BMAA as the culprit due to the low doses found in the cycad plant products consumed by the natives, such as cycad seed flour. However, upon evaluating other food sources in their diet, it was found that protein-associated BMAA in organisms that fed upon cycads, particularly the Mariana fruit bat, a part of the Comoro diet, were much higher, and explained how BMAA could reach necessary levels in patients to produce the disease state. I think I'll be avoiding the bat soup given recent trends. Our current understanding of BMAA and its effect on amino acid metabolism revolves around a protein incorporation hypothesis, in which BMAA is misincorporated in place of proteinogenic or canonical amino acids. We know that chronic BMAA exposure is associated with protein aggregation in vivo and in vitro. We also know that the enzyme allotyrinase synthetase has been linked with the misincorporation of BMAA due to the misacylation of amino acids in the aminoacyl pathway. It's during this pathway that amino acids for incorporation into protein are selected and marked for use, with allotyrinase synthetase being responsible primarily for acylating alanine. We also know that L-serine has been shown to be protective in vitro and in vivo based on primate studies that show decreased BMAA toxicity performed in 2016 and ongoing human clinical trials in which L-serine has been shown to slow the progress of motor neuron disease. What we don't yet know and hope to discover during this research is how L-serine is protective, and are those protective effects shared with L-alanine. We know L-serine is protective as it decreases protein aggregation and cell death in vitro and in vivo, but not by competition with BMAA for protein incorporation. Based on studies by Hahn and colleagues in 2019 into allotyrinase synthetase, there is reason to believe that L-alanine supplementation could be protective based on the evidence that BMAA is misincorporated for alanine in protein synthesis using isolated enzymes. The impact of this research is expanding with our understanding of BMAA production in our environment. It is well established that factors such as global warming and water eutrophication are contributing to an increase in blue-green algae blooms such as those tracked by Water New South Wales. BMAA has been proposed to be produced by most, if not all, cyanobacterial species by Paul Cox and colleagues in 2005. The implications of this theory have been seen in more recent studies, with BMAA being prevalent in food web studies of southern Florida, France, Sweden, as well as the Baltic Sea, wherein bioaccumulation allows higher concentrations of BMAA to reach significant quantities within food sources used for human consumption, such as fish and mussels. BMAA toxicity may not be limited to consumption either, with ALS incidents around a lake in New Hampshire in the northeastern US being found to be 25 times higher than the surrounding areas. The lake in question experienced frequent algal blooms, with recreational use and aerolization of BMAA from algal blooms being indicated as possible causes. My hypothesis is that BMAA modulates levels of protein amino acids in vitro and in vivo, with downstream effects on protein homeostasis, leading to protein aggregate formation. My investigation into the hypothesis is summarized by three aims. The first aim is to determine if L-alanine competes with BMAA for incorporation into protein using an LCMS or liquid chromatography mass spectrometer. Previous studies have not excluded exogenous L-alanine, which may have masked effects of BMAA on incorporation studies. This aim mirrors previous experiments evaluating whether L-serine competed with BMAA for incorporation. The method for aim 1 begins with cell culturing of an established cell line of SHSY5Y neuroblastoma cells in EMEM media. EMEM contains no alanine, serine, and proline supplements to allow for a control, as well as treatments with 0.01 molar alanine, serine, and proline. In this case, proline is used as an amino acid control. All cells are given a treatment of 1000 micromolar BMAA for 24 hours. The cells will be harvested with water and a few microliters from each well taken for a BCA assay for protein normalization later. Protein will then be precipitated from the samples using TCA and centrifuged, with the supernatant being discarded, as we are only looking at incorporated amino acids. The protein is rotary evaporated to remove liquid before moving to a hydrolysis vial, in which the oxygen is removed and supplanted with nitrogen to create an inert environment. The samples are then left in a 110-degree oven for 16 hours to finalize the breaking up of the protein into the constituent amino acids that are reconstituted in HCl to be run on the Shimadzu LCMS 8060, a triple quadrupole mass spec, giving us the relative abundance of amino acids alanine, proline, serine, and BMAA. The second aim is to determine if BMAA alters levels of all 20 protein amino acids in the cells upon exposure. The method for aim 2 starts with two 12-well plates of SHSY5Y, with one plate using EMEM as per aim 1 and another using DMEM, which contains exogenous sources of our protein amino acids, supplemented in aim 1. These two media are used to look at differences in the free amino acid profile based on growth media having reduced or supplemented amino acids. The samples treated with BMAA will again be with 1000 micromolar for 24 hours. Preparation of the samples for LCMS remain the same as before, however this time we are taking the supernatant fraction for analysis and preserving the pellet for a BCA assay for total protein. We are taking the supernatant fraction as it contains the free amino acids and not those incorporated into the protein fraction. The supernatant is subsequently freeze-dried to remove the liquid and to preserve the amino acids that are then reconstituted in HCl and run through the mass spec as per aim 1, although this time we'll be acquiring data for all 20 protein amino acids as well as BMAA. The final aim is to determine if L-alanine exhibits similar protective effects against BMAA-induced protein aggregation as L-serine by performing ubiquitin accumulation microscopy studies and a cytotoxicity assay. Part 1 of aim 3 will use the SHSY5Y cells in EMEM with supplementation in the second and third rows of serine and alanine at 0.01 molar respectively. BMAA treatment will also be at 1000 micromolar for 24 hours. Cells are subsequently washed with PBS and fixed with paraformaldehyde before incubation with AB7780 ubiquitin antibody and its secondary antibody with a conjugated fluorophore CF488 and a darbunuclear stain. These cells are then imaged under an Olympus BX51 fluorescence microscope and the images transferred to ImageJ software for quantitative analysis, where the fluorescence of ubiquitin is normalized to the cell number determined by the Darby stain. The second part of aim 3 is a flow cytometry cytotoxicity assay on the SHSY5Y cells that uses an Xn5 and propydium iodide to differentiate between healthy and apoptotic, necrotic, and dead cells. Plate 1 contains the controls outlined in the diagram, with the second plate having replicates of BMAA treatments at 1000 and 2000 micromolar for 24 hours. After cell harvesting, the reagents and Xn5 and propydium iodide added in the required samples and run through the BD LSR Fortessa flow cytometer to determine cell viability post-BMA treatment as a percentage of the total number of cells. This is possible due to an Xn5 and its conjugated fluorophore binding to the phosphatidylserine that is usually limited to the inner surface of the plasma membrane bilayer, however is exposed to the surface during apoptosis, and propydium iodide is a dye that is excluded by viable cells as they have an intact membrane. In summary, if alanine can compete with BMAA in protein incorporation, we will have evidence that BMAA can be competed with and can develop ways to prevent non-native proteins being generated. If alanine is protective, then it can be evaluated in the same manner as L-serine, and research into its therapeutic use can be conducted. If BMAA alters amino acid levels and affects protein homeostasis, we can use the specific amino acid profile generated to elucidate the exact mechanisms and pathways through which BMAA exerts its neurotoxic effect. I'd like to take this time to thank my supervisors Ken Rogers and Alan Fays, the technical staff Luke, Mercedes, and Sarah, and finally the HDR students of my lab group, Carly, Jake, Joel, Lisa, and Siobhan. Thank you.